Hi, my name is Dr. Richard Doveston and I'm a lecturer in chemical biology within the School of Chemistry at Leicester Institute of Structural and Chemical Biology. My research has a specific focus on using molecular glues to modulate protein signaling pathways, but in a much broader sense it involves designing, making and evaluating new molecules that might be effective as drugs themselves or might be useful as tools with which to study the biology behind disease. Drugs typically work by targeting biomolecules in our bodies called proteins. The interaction of a drug with its protein target will directly affect that protein's ability to carry out its function, and thus we see a biological response. We can crudely classify drugs as either biologics or small molecules. Biologics are usually biomolecules themselves and are exemplified by the very successful antibody therapies that have emerged in recent years. However, small molecules remain to be the mainstay of the pharmaceutical industry's R&D effort, and these drugs are the ones that are most familiar to us. They are usually organic molecules like aspirin, shown here, or simvastatin, shown here, which is a highly successful drug used to lower cholesterol levels in our bodies. Small here is really a relative term, in that what we mean is the drug molecule is small relative to its protein target. So within the red square here we can see a drug binding to a very specific pocket of a much larger protein biomolecule. Although small compared to the size of its protein target, the intricate and specific interactions of a drug with the protein are absolutely fundamental to it being effective and selective. And if we zoom in, we can see in great detail those interactions taking place. Despite the volume of research that's been given over to small molecules over the years, we do tend to stick with what we know. And the pie chart on the right here illustrates this. Just over 2/3 of all known small molecule drugs target just 4 different types of protein, which is really a drop in the ocean compared to what's possible. So the big question therefore is, how can we expand this portfolio below the low-hanging fruit, the easy targets, and thus develop new drugs for diseases that are currently incurable, or perhaps circumvent issues of drug resistance? One possible approach is to tap into and target a vital communication network within our bodies that relies on the interaction between proteins, or protein-protein interactions. There are over 300,000 PPIs in our bodies, or estimated to be, and many of these are implicated in disease. So if we can design a molecule that interrupts that interaction, or perhaps helps that interaction, we might see a favourable biological response. However, they are very challenging drug targets, whereas in the traditional sense which we just discussed, drugs are targeting a very specific pocket, very well defined pocket within a protein target. Protein-protein interaction interfaces are very large surface areas. It's very difficult to design drugs that selectively target those. That said, the field of protein-protein interaction inhibition has been highly successful and productive in recent years, and now delivered protein-protein interaction inhibitors into the clinic, and many more are currently in clinical trials. However, we could expand our druggable universe even more if we didn't just focus on inhibition, but also thought about stabilizing protein-protein interactions. So perhaps there's an interaction that's really important for our body's fight against disease. Can we in some way supercharge this by using a small drug molecule as a glue, as a molecular glue? So on the right here we have a protein in grey called 1433 and a protein partner that binds to it called PMA2 in green, and at the interface of those two proteins we see a molecular glue bound.